Hello everyone, I am Nivedita Jaina from Ambika Prasad Research Foundation. Welcome to all of you to our channel Biodiversity and Conservation. So today we are going to discuss about the phytochemical training. So let's start. Phytochemical screening techniques involve botanical identification, extraction with suitable solvents, purification, and characterization of the active constituents of pharmaceutical importance. And this screening confirms the presence of phytoconstituents like alkaloids, flavonoids, glycosides, and lignins like biochemical compound. And the phytochemicals have great antioxidant potential and are of great interest due to their beneficial effects on health of human beings, and they give immense health benefits to the consumers. And also the phytochemicals are the natural chemicals present in plants which helps to protect plants from germs, fungi, bugs, and other threats. The phytochemicals have two categories. One is the primary and another is the secondary constituents. And the primary constituents include carbohydrate, amino acids, proteins, lipid, and chlorophylls. And secondary constituents are alkaloids, tannins, saponins, flavonoids, steroids, phenols, and reducing sugar. And the phytochemical analysis has commercial value and great interest in pharmaceutical companies for the production of the new drugs for curing of various diseases. Here is the phytochemical analysis methods for different phytoconstituents present. So for the carbohydrate, it is the molecular test, and protein and amino acids, it is the anhydrous, burette, or Miller's test. For alkaloid, it is here drag and drops test. Then for tannin, it is ferrichloride and saponin foam or fort test. Yeah, for flavonoids, lead acetate and alkaline reagent is used. And for reducing sugars, it is Fellings or Benedict's reagent test occur. For terpenoids, it is Salcoacetic test. For phenol, ferrichloride and lead acetate test. And for phytosterols, it is here Lieberman and Borcher's test. For the detection of carbohydrates, first dissolve 2 gram extract in 5 ml distilled water and filter it. And the filters were used to test for the presence of carbohydrate. So in Molly's test, the filtered was treated with 2 drops of alcoholic alpha naphthol solution in a test tube. Then second, and then add concentrate sulfuric acid from the side of the test tube. If development of a violet ring at the junction of two liquids confirm the presence of carbohydrates. And the detection of carbohydrates, first dissolve 2 gram extract in 5 ml distilled water and filter it. And the filtrates were used to test for the presence of carbohydrates. So in Molly's test, the filtrate was treated with 2 drops of alcoholic alpha naphtha solution in a test tube. And then second, and add concentrate sulfuric acid from the side of the test tube, and the development of a violet ring at the junction of two liquids confirmed the presence of carbohydrates. And for the detection of proteins and amino acids, so the test solution was added with few drops of Miller's reagent. When warmed, a white precipitate is formed which changes to a brick red or disappears. Then it indicates the presence of proteins and amino acids. And in ninhydrins test, the test solution was added with ninhydrins reagent and boiled for few minutes. And the formation of blue color indicates the presence of amino acids. Then for the detection of alkaloids, the individual extract is dissolved in dilute hydrochloric acid and filtered, and then the filtrate was further tested with the potassium bismuth nitrate solution, which is the Dragitrof's reagent, and then the formation of orange red precipitate indicates the presence of alkaloids. And for the detection of tannins, First, take 0.5 gram of the dried powder of plant and then boil 0.5 gram sample in 20 ml of water in a tester and then filter and the above mixture and to the above mixture add few drops of 0.1% ferric chloride and then the development of a brownish green or blue black coloration indicates the presence of tannins. Then in alkaline reagent test for the detection of flavonoids, the extract was treated with sodium hydroxide solution, and if the formation of an intense yellow color indicates the presence of flavonoids, and in lead acetate the taste, if the extract was treated with lead acetate solution, and if the formation of yellow precipitate is occurred, then it indicates the presence of flavonoids. And for the detection of saponins, it is in for the test. The extract was diluted with distilled water to 20 ml and second in a graduated test tube for 15 minutes. And the formation of 1 cm layer of foam indicates the presence of saponins. And in foam test, a small quantity of the extract was taken with 2 ml of water and the persistence of foam produced for 10 minutes indicates the presence of saponins. And for the detection of terpenoids, it is here the sarcosic test in which the extract was mixed with 2 ml of chloroform solution and added and concentration sulfuric acid to form a layer, and then a reddish brown coloration of the interface indicates the presence of terpenoids. And in Benedict's test for the detection of reducing sugar, the extract was or the filtrate was treated with Benedict's reagent and boiled in a thermostatic water bath for 5 minutes, and then the formation of an orange red precipitate indicates the presence of reducing sugars. In ferric chloride test for the detection of phenolic groups, the extract was treated with 3 to 4 drops of ferric chloride solution, and then the formation of bluish black color indicates the presence of phenolic groups. And in for the detection of phytosterols, the Lieberman Virchow test is defined in the chloroform extracted solution is treated with few drops of acetic anhydride, and then it boiled and cooled. Then the formation of green color solution indicates the presence of phytosterols. I hope this discussion would informative to all of you. Thank you.